Howdy, today on Flipping Science we're looking at hydrogen and ion dipole bonding. So some substances that are molecular have higher boiling points than you might think of, and this is because of hydrogen bonding that occurs between the molecules. So hydrogen bonding occurs in molecules where you have bonds between hydrogen and fluorine, oxygen and hydrogen, or nitrogen and hydrogen. So here we have some examples. So over here we have ethane, which doesn't have any of those bonds. It's only got carbon-hydrogen bonds, or a carbon-carbon bond. Its boiling point is minus 88 degrees Celsius. We take something with a very similar molar mass, methanol. Methanol, it has carbon and hydrogen bonds, but it also has a carbon-oxygen, but really an oxygen-hydrogen bond, and that's the main thing. So because it's got those oxygen-hydrogen bonds, it can have hydrogen bonding, and that increases its melting and boiling points. So what happens is in molecules with those bonds between hydrogen, fluorine, oxygen, hydrogen, nitrogen, hydrogen, what happens is you've got hydrogen that's covalently bonded to a very electronegative element, so fluorine, nitrogen, or oxygen. So these are all very electronegative, and what that means is in the bond, these are pulling the electrons closer towards them, so they get a very strong partial negative charge. What happens with the hydrogen atom is because the electrons from the bond have been pulled towards the electronegative elements, it gets a very strong partial positive charge. It's almost an exposed proton. There's very little interaction with the electron. So there is some, but not much. So what that means is you get a very big difference in polarity between the hydrogen, fluorine, oxygen, hydrogen, or nitrogen, hydrogen. So that generates the hydrogen bond. So if we have a look at example here in water, we can see in water we've got oxygen and hydrogen bonds. Each hydrogen is going to be delta positive. It's got a partial positive charge, and the oxygen is delta negative. So you get a very big difference in electronegativity, and what that means is that the oxygen from one molecule will be attracted strongly to the hydrogen of another water molecule. And what this means is that there's this very strong force of attraction between the water molecules, and this does things like increase its melting and boiling point. So to say that again, you get a very strong electrostatic attraction between the delta positive hydrogen and the delta negative fluorine, nitrogen, or oxygen in the nearby molecule. So what happens is you get this matrix of molecules where each molecule is attracting several other molecules nearby with very strong electrostatic attraction. And this requires a lot of energy to break those electrostatic bonds that are occurring between the molecules. And that's why the melting point and boiling point increases with these molecules. We represent the hydrogen bond by using dotted lines between the delta positive and delta negative molecules, parts of the molecule. So hydrogen bonding is very important for us. It forms bonds between the oxygen-hydrogen groups in water, and what that means is that water at room temperature is a liquid rather than a gas, and that's very important for us as animals. It forms between the nitrogen-hydrogen groups in ammonia, amides, amines, amino acids, proteins, synthetic polyamides, and DNA. What this means is that you can have long strands of molecules in the polyamides and DNA and proteins that can be attracted between each other, and that can cause folding, or it holds the strands together more, and that leads to important properties. So in proteins, bonding between chains leads to different shapes in the proteins, and the shape of a protein tells you its function. So in DNA you have two strands of nucleotides, and they're being held together with hydrogen bonding, and that hydrogen bonding between the DNA strands is what keeps the DNA together, and it makes it very, very stable. But it also doesn't require really that much energy to break, so you can separate out the DNA. And that means you can do things like replication, which is very important. You can also form hydrogen bonds between a hydrogen and the carbon double bond oxygen groups in carboxylic acids, amides, amino acids, aldehydes, ketones, proteins, synthetic polymers, and between DNA as well. So if we have a look at this, here we have an amide group in one protein strand. We've got a C double bond O here, and we're getting hydrogen bonding between that and a nitrogen-hydrogen bond from the next nylon molecule. This is over here. So we're getting attraction between the amide groups from one strand to another, and that holds the nylon together. And that means the nylon has a fairly high melting and boiling point. If we look at DNA over here, we can see, so between adenine and thymine, these are two nucleotides. The adenine has a nitrogen-hydrogen bond over here, and over here on the thymine we have a carbon double bond oxygen bond, and we get attraction between those. Like I said, that's what holds these together in the DNA strand, the double helix. And that's very important biologically. So hydrogen bonds are important because they're the strongest form of dipole-dipole interaction. So that means they're very good at holding molecules of substances with hydrogen bonds together, increasing melting and boiling point. But that doesn't mean they're as strong as covalent bonds where you're sharing electrons between atoms. The reason why they're the strongest form of dipole-dipole is because of that big electronegativity difference between the delta-positive hydrogen and the delta-negative nitrogen, oxygen, or fluorine. So there's a really big electronegativity difference there, and that leads to a very strong secondary bond between the molecules. The other thing is that hydrogen is very small, so it can get very close to the delta-negative atom in the other molecule. Now, it's pretty tricky to compare secondary interaction strength unless you control for a few things. You need to control for shape and you need to control for molar mass, because both of those will affect the strength of any secondary interaction. But if you've got hydrogen bonding, the deal is that if you have a similar molar mass and a similar shape, if something can exhibit hydrogen bonding, then we'll have a higher melting and boiling point compared to the other molecule. So here's some examples. So here's SiH4 down here. It's got a molar mass of 32, which is similar to methanol that we have over here. Kind of a similar shape. We've got it's a tetrahedral structure over here, and we've got a tetrahedral part at least to the methanol. The secondary bonding you're going to get in SiH4 is only going to be dispersion forces because it's a non-polar molecule. So we do have a delta-positive, delta-negative interaction in the bond between the silicon and the hydrogen, but because it's tetrahedral, that's all cancelled out. So the delta-positive here, here, and here, the average of those positions is in the middle of the silicon atom. So the center of partial positive charge is the same place as the center of partial negative charge. So that's not going to we're not going to get anything other than dispersion forces between the molecules. Whereas with methanol over here, we have the oxygen-hydrogen bond, so we're going to get hydrogen bonding between the methanol molecules, and that's the reason here why SiH4 is minus 112 degrees Celsius for its boiling point, whereas methanol is 64.5 degrees. So there's a big difference in boiling point there. Ion-dipole bonding is a little bit different. It occurs where you have an attraction between a fully charged ion and a molecule that has a dipole. So here's a nice picture of it. So we've got a fully charged anion. We have a polar molecule with a delta positive and a delta negative end. The positive delta positive end is going to be attracted to this anion here because it's got a negative charge. And similarly, if we have a positively charged cation down here, the delta negative end of the molecule will be attracted to it. So if you have a chloride ion in solution, we're going to get bonding between ion dipole bonding between the hydrogen parts of the water molecule, which have a strong delta positive charge, and the negative charge of the chloride ion over here. So there's going to be strong attraction between them. Over here with the sodium ion, it's going to be the opposite. So the oxygen is delta negative in a water molecule, and all those oxygens from water molecules around are going to be attracted to the cation, the sodium cation. The reason why it's so strong is because you have a full charge. It's not just a partial charge, which is what delta means. It's a full charge on your anion or cation. And this doesn't have to be a single charge either. You could have a double negative charge, or a double positive charge, or a triple positive charge. So what that means is you're going to get a lot of polar molecules surrounding your anions and cations. And that makes them very readily soluble, for example. So salts are very soluble in water, usually because the anions and cations form ion-dipole bonds with the water, and they can make their way into the water matrix. So they break up the hydrogen bonding between the water molecules, and you get bonding between the water molecules and the anions and cations. So let's do an example question. So here we have dihydroxy acetone. It's a simple carbohydrate. We've shown its structural formula. Explain why dihydroxy acetone is soluble in water. So this is a question from organics, but it matches up to what we've been talking about in terms of hydrogen bonding. So to be soluble in water, you need to be a fairly small molecule, and you also need to be able to make some kind of interaction with the water so that the water dissolves you, essentially. So you can make your way between the water molecules and the water matrix. So here, if we look at dihydroxy acetone, we've got two hydroxyl groups, so OH groups, and we've also got a C-double-bond-O group, and each of those can form hydrogen bonds with water molecules. So because it's a small molecule that can form lots of hydrogen bonds with water molecules, it's going to be readily soluble in water. Here's another question. It says Diclofenac is an anti-inflammatory drug used for humans and animals. We're given the structure. It says Diclofenac is taken in the form of a sodium or potassium salt. Explain why the salt is more soluble in water than the molecular form. So the first thing I'm going to do is I'm going to change change it so you can see what the sodium salt would look like. So what we're going to do is we would react it with, say, sodium hydroxide, so this carboxylic acid group. We get a negative at the oxygen, and then we'd have Na+. So we'd have the sodium ion attracted to the negative oxygen over here from the carboxylic acid group. So now that we've got that, we can talk about why it would be more soluble in water than the molecular form. The molecular form is just that with the hydrogen. So when you chuck that in water now, the sodium ion is going to ionize, the sodium ion will be floating around in solution, and we're left with a fully charged, negatively charged oxygen atom down here. And what that means is we're going to get ion dipole bonding between the negative ion here and the positive hydrogen parts of the water molecule, so the partial positive hydrogen parts of the water. So we're going to get ion dipole bonding between those down here, and what that means is that will make the whole thing a lot more soluble because of that ion dipole bonding. So it's a fairly large molecule, but because we have that ion dipole bonding occurring here, that's enough to overcome the hydrogen bonding between the water molecules, and that makes it more soluble. So today on Flipping Science, we looked at ion dipole bonding and hydrogen bonding. That's it for Flipping Science today. See ya.